Let me welcome everybody to the May 28th meeting of GLIMPRINT. We have a talk today from Scripps about using graph neural networks to enhance drug repositioning. This is a topic that is of significant interest to many people in our audience. We're very pleased that Carolina Gonzalez Cavazos has agreed to speak to us today. As always, Tom Helikar, myself, Jim Sluka, Lorenzo, and Ellen are available to take your questions, comments, concerns. Please don't hesitate to reach out to us if we can do a better job. We have a variety of communication channels. They work better if you use them. I would encourage people to help us get the word out, especially about the YouTube archive of the videos. As always, this meeting, like all of the meetings, is recorded, live streamed, and made available in edited form later as part of our talk archive. That talk archive has more than 160 talks in it now, covering all aspects of immunology, modeling, digital twins, and we would love to have that get more use. If you have a publication that you would like to share, it can be put up on the IMAG wiki page, which has been going from the beginning of this group. Coming up, we also have a variety of seminars. June 11th, we have a talk on single-cell multimodal data integration for understanding clinically relevant states. We have a talk from Purdue University on June 25th. As always, you've heard me say it before. If you would like to speak, suggest yourself, suggest your students or your postdocs, anything that's relevant in this broad area. It could be on immunology, it could be on infection, could be on multiscale modeling of any kind, could be on data collection methods, data analysis methods, drug discovery, experimental techniques, biotechnology, anything that affects this very broad space of computational immune techniques. And if you've already spoken and you have something else to say, don't feel shy about giving a second talk. We've had many people come back and give multiple talks over the years. We're always happy to have you do that. You can either email Lorenzo or me with your ideas. We'd love to hear what we can do to make this series more appealing to people in the community. So today we have a talk on enhancing drug repositioning with interpretable graph neural networks on biomedical knowledge graphs. I had heard this talk in San Diego and been quite impressed by the work, and I thought it would be a topic that would be of great interest to our community. These are techniques I think that we could use very extensively in the work that we discuss here. And I appreciate Carolina being willing to come to tell us about her work.
Hi, everyone. My name is Carolina. I'm a staff scientist at the Scripps Research Institute in San Diego, working in Professor Andrews' lab. Thank you so much for having me today. I'm really excited to present our work on biomedical knowledge graphs for drug discovery. And I guess I can start presentation now. I want to start this presentation mentioning that this work is motivated by the current challenge of developing new drugs. The process of discovering new drugs is extremely time-consuming and expensive. These figures over here shows that getting a drug to the market takes between 12 to 16 years, and the average cost to develop a new drug from scratch is between $1 to $2 billion. But what I think is more concerning about developing new drugs is that the success rate is really, really low. As you can see, only a few drug initiatives will successfully make it to the market. So one promising solution to these challenges is drug repositioning, which basically involves finding new therapeutic uses for approved drugs that are already out on the market. So since these approved drugs have already gone through some safety and efficacy evaluations, repositioning them reduce the development costs and the timeline to bring them to the market, which of course accelerates the process of identifying novel treatments for diseases that currently lack therapies. So there is a growing interest from the pharmaceutical industry and the research community in developing new strategies for drug repositioning. There are a bunch of computational and experimental approaches that have been developed for drug repositioning. But there is one fundamental challenge for this. All the biomedical knowledge is essentially fragmented. All the information we know about drugs, genes, pathways, and diseases is spread across many disconnected databases. And these databases, of course, sometimes they don't communicate with each other and makes understanding the biological relationships that are critical for drug repositioning really difficult. And we can take an example like imatinib. We know imatinib is used to treat leukemia, but if we want to understand how imatinib works, we need to search across multiple databases. For example, Drug Bank will tell us that imatinib treats leukemia, and then we have to go to gene ontology to understand how imatinib inhibits the BCR pathway. And then other databases can tell us that the same gene is associated with leukemia. And then other resources can tell us that how that gene is associated with other downstream genes. So even that each of these relationship is really valuable, all this information is scattered across separate databases. And we only see fragments of a big picture, right? So how exactly can we overcome this fragmentation? So this is where biomedical knowledge graph come into play. Knowledge graphs are a technology that is designed to organize and connect information. So instead of storing facts in isolation, they represent knowledge as a big network of entities and the relationship between these entities. And when we apply knowledge graph to biomedical data, we can start having facts about drugs, disease, and genes, which were isolated in many different databases into one single place. So now information about drugs, diseases become nodes, and the relationship between these biological entities, such as treats or inhibits, becomes an edge. So now what was fragmented before in many different databases starts to become a big network that allows us to see patterns and connections that we wouldn't be able to see in one single database. And then you can start imagining that we can scale our biomedical knowledge graph. Instead of just having few nodes or one drug mechanism, you can capture thousands of drugs and diseases and other biological entities, all one interconnected in one comprehensive network. And once we have this big network, we can see how drug repositioning starts to become really powerful. We can take imatinib as an example again. We know that imatinib treats leukemia, but when we incorporate other databases, we can see that imatinib is associated with other genes. For example, we see that imatinib inhibits other kinase genes, which are associated with the activation of mast cells. And this activation is associated with the asthma, right? So the question that arises from seeing these patterns is whether imatinib can be used to treat asthma. And again, this is where drug repositioning starts to become really, really powerful because we can take the knowledge from the knowledge graph that is encoding information from different data sources, and we can start seeing hidden patterns and start to discover novel connections. So using biomedical knowledge graphs for drug discovery essentially ask to see these indirect connections and start to predict new connections for a given drug in a given disease. But then now the challenge is how do we analyze these big networks? Because again, it can incorporate thousands and millions of biological entities, and it can be really challenging to analyze patterns and predict novel connections, right? So we need a simpler way to represent our knowledge graph. We need to basically represent and translate the structure of the graph into something simpler that a machine can learn from. And this is where graph neural networks come into play. The idea is very straightforward. We take our knowledge graph with drugs, diseases, genes, and all the other biological entities, and we map every node or every entity into an embedding space in a way that we preserve the original structure of the graph. So the idea of graph neural networks is to learn an embedding representation for each node, capturing the role of every node within the big network. So the intuition is that those nodes that share similar neighbor will end up close together in the embedding space. So for example, we see that drug A and disease A, which we know that are known indication, will share similar genes and similar pathways, and the GNN will allow us to learn a function that will map them close together in the embedding space. And for drug repositioning, that is really useful because if a drug and a disease that share similar neighbors, like similar genes and similar pathways, their embedding will naturally end up close together in the embedding space. So we can see here like drug A and disease C that doesn't share a connection in the original knowledge graph. Graph neural networks allow us to capture the neighborhood and place it close together in the embedding space. And so that proximity in the embedding space allows us to predict novel drug disease connections. All right, so these graph neural network architectures can be really impressive, predicting connections that we know that are true and also predicting novel connections. And the problem here is that graph neural networks can be seen as a black box models. We don't really understand why they make certain predictions, right? So in drug discovery and many other research areas in biology, we are interested on knowing more than just a link between a drug and a disease. We really need to understand what is the biological mechanism behind these predictions. What are the paths and what are the networks that connect a drug and a disease? But what these models are missing is those biological paths, those explanations that make that are very useful for domain experts to make better informed decisions. So having this challenge in mind, the main goal of this project was to implement a knowledge graph link prediction framework that not only predict drug disease connections, but also provide interpretable explanations for these predictions. Now we had to build this framework, and the first question we had was, where do we even start, right? Because as you can imagine, we have thousands of possible drug disease combinations that we can evaluate, but evaluating all these combinations is simply not feasible, right? It will take a lot of time. So the first thing that we ask, and how do we tackle this problem, we thought about narrowing down the possibilities in a smarter way. And the intuition for us was like, it was like a very simple biological observation. We know that similar drugs tend to work through similar biology. For example, in this very simple figure, we can see that we have drug A, drug B, and drug C treating their respective diseases, but they all share the same genes and the same pathways and the same cellular targets. And we can borrow that reasoning for a new drug of interest. And as you can see, we can start with these drugs and take that knowledge and use those paths, as we show on the right, and reuse them to identify more promising disease candidates. So now, instead of evaluating thousands of combinations for a given drug, we can only focus on the most biological relevant candidates. So now we can use similar drugs that have like similar biology and target the search in a more smarter way. So this idea of learning from similar cases is not new at all. There is a classical artificial intelligence approach called case-based reasoning that is built on this exactly same intuition. Instead of solving problems from scratch, you can look at problems. You can look at similar problems that you have solved before and use that knowledge that has worked in the past for a problem that you have right now. And I think a good way to understand case-based reasoning and to understand the intuition is through a really simple analogy where you can think about a chef that is trying to recreate a pasta dish. And instead of starting from scratch, the chef can think about similar recipes, like the ingredients, the techniques, the proportion that worked in the past, and apply that knowledge for the new dish that he's trying to recreate. So that's exactly how case-based reasoning works. You don't need to start from zero. You can learn from what has worked before. And we can formalize case-based reasoning in a simple three-step process. We first have a problem and we retrieve similar cases. Once we have the similar cases, we can reuse the solutions from those similar cases. And then finally, we can evaluate if the solutions were actually helpful for the problem that we're trying to solve. So using this three-step process, we apply that intuition for our problem. And this is just a summary of all the steps of our framework. I will go through one step at a time in the next slides. But as you can see, we are following the three main case-based reasoning steps. So we start with a drug, and we are interested in finding new possible disease indications. And the first step is to retrieve similar drugs, and I will go into that in a minute. As you can see, we are not only retrieving similar drugs, but we are also retrieving from the knowledge graph the mechanisms that explain a drug to a given disease. And then we could take these paths and reuse it apply it and reuse it for a query drug that we're interested in. As you can see, it allows us to navigate the knowledge graph and arrive to possible disease candidates. And then finally, we need to evaluate which of these candidates in the embedding space is more likely to be a more valuable or more likely candidate for the drug that we're interested. So again, with this framework, we are not just producing novel predictions, but we have access to the biological explanations that allow us to understand the predictions. All right, so we'll go through the three steps. And the first step is to retrieve similar drug cases. How exactly can we define the similarity between drugs? We do it in a very simple way. The first thing is that we need to represent all the drugs that we have in the knowledge graph. And so for each drug that we have in the knowledge graph, we extract the direct neighbors. So you can see that a drug can be connected to the cell, to a symptom, to a biological processes, and we encode that knowledge into a vector. So each position in this vector represents one entity in the knowledge graph. And if a drug has a connection to that given entity, you assign one, and then if there is an entity on the knowledge graph that is not connected to that drug, you assign a 0. So you have a big vector for your drug, and you basically do the same for all the drugs that exist in the knowledge graph. So we create what we call it the memory of drugs. As you can see, every row is one drug, and it has like one vector representation for every drug. And then when we want to identify the most similar drugs, we essentially calculate cosine similarity. So we have a drug of interest and calculate cosine similarity with every drug. And the intuition here is that the cosine similarity, the score between a given drug and the most similar drug will be high because they share similar neighbors in the knowledge graph. So once we define the similar drug cases through this simple similarity metric, we can reuse the drug solutions. So for this, I'm just using an example so we can visualize what that means. So propranolol is a drug that was approved by the FDA for hypertension. And what we see here is that using the similarity score I just mentioned, we are getting this top 3 similar drugs, which are also drugs that are used for heart-related issues. So again, once we have these drug solutions, we can look at the mechanistic paths for each of these drugs. In this case, I'm just showing clopidogrel. But what we do is to take clopidogrel and navigate the knowledge graph and see what are the paths that connect clopidogrel to acute coronary syndrome in this case, because that's the disease that clopidogrel treats. And of course, there are like certain parameters that you can decide over here, like what are the number of paths that you select, the number of neighbors, and so on. But essentially, you do the same for every drug that you define as similar, and get the paths of them, right? The idea is that take those paths and generalize them as metapaths. So meaning that we abstract the information into a higher level, so we can see patterns between the similar drugs. So now, instead of having specific entities, we have a sequence of node types. So we see that a drug is affected to a gene is associated to a pathway in biological processes. And so we abstract these patterns across drugs in a way that they are more generalizable or more applicable to a query that we are trying to solve. So let's say that we abstracted all the information into metapaths. So once we have these metapaths, we can go back to the query, to the drug of interest that we're trying to solve, and we can reuse those metapaths for propranolol in a way that we can start navigating the knowledge graph. So they kind of serve like as a guide to navigate the knowledge graph in a smarter way, or in a way that we can restrict the navigation. And that allows us to arrive to potential candidates, including the correct answer for propranolol, hypertension, and other candidates, possible candidates such as Parkinson in this case. But then, as you can imagine, sometimes we don't arrive to the correct answer, or we arrive to candidates that are less likely to be a good candidate, such as bacterial infection. So the last step is to identify which of these candidates are more promising. So now we have identified the candidates, right? And we need to understand which one are more likely to be a good candidate. And for this, we are going to use a graph neural network strategy. So we have the subgraph from the similar drug cases. We see clopidogrel connected to acute coronary syndrome, and we see the subgraph from propranolol connecting hypertension with Parkinson and bacterial infection. We generate embeddings with a graph neural network strategy. So we see that we generated an embedding for acute coronary syndrome. And we do the same for propranolol. We ended up having representations for all the candidates. And then using the case-based reasoning hypothesis, where similar cases should have similar solutions, what we expect to see in the embedding space is that the solution for our similar case should be similar to the solution of the drug that we're trying to solve. So we expect that acute coronary syndrome to be close to hypertension in this case. And then naturally, other similar associations, such as Parkinson, will also end up close together in the same embedding region. So as you can kind of see in this figure, we see that Parkinson is sharing some mechanisms with hypertension, which is the correct answer. And then other candidates, such as bacterial infection, which share less mechanisms with the correct answer, will end up far from this embedding region that we know that is true. So basically, you can rank the most likely or most promising candidates with a simple cosine similarity with the correct answer. And this is just the learning objective that we use during training. And the idea is that we are basically training our model in a way that we maximize the similarity of correct answers and minimize the similarity of a correct answer and an incorrect answer. So taking this example, what we basically do is to maximize the similarity of hypertension and acute coronary syndrome, and minimize the similarity of acute coronary syndrome and bacterial infection. And I think that was basically the overview of the pipeline. Again, case-based reasoning is a 3-step process. There are like a little bit more of technical details behind each step, but I think I really hope the intuition came clear. But before going into the results of our model, I just wanted to take a moment and talk about our collaboration with the EveryCure project. I don't know if people have heard about it. It's a really cool project. We started collaborating with them, I think like 2 years ago. EveryCure is a nonprofit organization that was founded in 2022 that also focus on identifying novel uses for drug repositioning candidates through different computational approaches. But what I think EveryCure is really meaningful is the story behind it, and it really reminds us why drug repositioning strategies are really powerful. So Dr. David is one of the founders of EveryCure, and he was a medical student in 2010 when he was diagnosed with a really rare immune condition called Castleman disease. This is a condition where basically the lymph nodes are overactivated and produce a lot of immune cells. And he mentioned that he nearly died 5 times. And every time that the doctors told him that he was about to die, they told him the same thing. There is no treatment options. Developing a new drug will take a lot of time, a lot of money, and there's nothing to do essentially. And so with no treatments, he decided to analyze his own blood samples and his own lymph node samples, hoping to find like something that the medical team didn't see. He actually found that the mTOR pathway was hyperactivated, and he did some like research on the literature and found there was a drug that was originally used for organ transplant that was actually inhibiting this mTOR pathway. So the biology made sense. He talked with the doctors and he started treatment, and he has been stable since then. And I think the story makes perfect sense of why drug repositioning is really powerful. And after that, he decided to found EveryCure. And again, the mission behind EveryCure is exactly this idea. We want to find novel treatment for existing drugs. So yeah, we started collaborating with them a couple of years ago. They basically provide us with a unified knowledge graph that integrates a bunch of open source biomedical databases. So the big unified knowledge graph that they are working on essentially integrates other knowledge graph, other 3 big projects that you might hear about, like RTX-KG2, Robocop, PrimeKG. Each of these big knowledge graphs already integrate other data resources that you can see over here. And this big unified knowledge graph incorporates 11 million nodes and around 80 million edges. So this knowledge graph is essentially the model that we were using to train the case-based reasoning pipeline that I just showed you. And there are 3 important elements of this knowledge graph that we were using to train and evaluate our model. Basically, we use the drug disease treatments from the FDA as ground-truth positives. So all those drug disease elements that we know that are approved, we are using them to train our model and use it for ground-truth positives. We also have access to ground-truth negatives, meaning contraindication. So sometimes we know that a drug is contraindicated for a disease. We use that to tell our model, like, don't learn from them. And then we also have access to something called off-label data. These are basically real-world drug repositioning cases. So right now there are some drug cases that are used for certain diseases. They're currently not approved by the FDA, but they are well documented. So this data set is really powerful because now we can train our model with ground-truth positives and ground-truth negatives and use the off-label as something that the model never seen and see how we are performing with them. I think that was pretty much about the summary of the data sets we are using for this project. As you can imagine, there is a lot of work behind building all these resources. I think the Every Cure has done a lot of work curating them. But for this project, we are using what is already available and ready to use, essentially. So coming back to the case-based reasoning framework we implemented, as I explained earlier, the main goal of is to follow the case-based reasoning hypothesis, where similar cases should have similar solutions, so they should end up close together in the embedding space, and cases with different solutions should not be similar. So in the context of graph neural network embeddings for ground-true positives and ground-true negative diseases, the idea is the same. We want the model to place ground-true positive example close to each other, and we want incorrect or the ground-true negative pairs to be pushed further away from this embedding region. And this behavior is driven by the learning objective that I just mentioned. And what these plots are showing is basically how the model is actually separating ground-true positives and ground-true negatives during training. The x-axis just represent the training steps over time, and the y-axis just represents the cosine similarity between the ground-true positives and the known similar drugs. So as you can see on the left, the ground-true positives starts to become closer to 1, meaning that the cosine similarity, the cosine similarity starts getting closer to 1. So similar entities starts to become more similar to each other. And then the model makes the ground-true negatives to start getting closer to -1, meaning that they are less similar in the embedding space. So yeah, this is just basically to show you how the training process works. But then now looking into the embedding space and to visualize a little bit more how the model is doing, what I'm showing you here is the embedding space for ground-true positives and ground-true negatives in a simple PCA. As you can see, the separation is not clear, it's very messy. And part of the reason why this separation is not trivial is because sometimes diseases can be represented as ground-true positives or can be represented as ground-true negatives. So really the separation in the embedding space is really hard to do. So globally, the embedding space looks noisy and overlapping. But when we look at a specific query example, so each of these is one drug case. And again, the red dots represent ground-true negatives and the blue ones ground-true positives. We can see very clear that the model is separating them in the embedding space. So even that in the global embedding representation, everything looks really messy. When we look at the local neighborhood, we see that there is much like a structure and a separation. We basically measure this distance between ground-true positives and ground-true negatives in a more systematic way. And what we see is that the similarity between ground-true positives and the similar drugs is higher, and the similarity between ground-true negatives and the similar drugs is less similar. All right, so now, as I mentioned, we're really interested on evaluating the off-label data, which again, this represents drugs that are currently used as drug repositioning cases. They are not approved by the FDA, but they are very, very well documented. We didn't use it for training, only for evaluation. And we were wondering if they were behaving more like a ground-true positives or more like a ground-true negatives. So similar to the previous visualization, each of these represent one drug case, and it's probably a little bit hard to see, but again, we have ground-true negatives, ground-true positives as blue, and the orange dots represents the off-label data. And what I'm trying to highlight here is that we do see in this visualization that the off-label data is really close to the ground-true positives. I think it's a little bit hard to see, but if you pay close attention, you see that the orange dots are closer to the blue ground-true positives. We did a systematic evaluation of this distance. So what is the distance of ground-true positives and the off-label? We do see that the similarity is higher for ground-true positives and off-label. And the similarity of ground-truth negatives and the off-label is a little bit more spread, so they are less similar. So yeah, we were really impressed that our model was able to place the off-label diseases or the off-label cases more similar to the neighborhood of the true indications. Yeah, this is just like another metric. So basically, for every drug that we have a ranking list of possible diseases. And with this metric, the hits@K metric, we are basically evaluating the percentage of diseases that appear at the top K prediction. So from the list, we ask the percentage of diseases that appear at top 1, top 2, top 3, and so on. And so this curve is just showing us that overall ranking, the top ranking, we do see that the ground-truth positives is outperforming the contraindications. So we do see that contraindication is way lower than the ground-truth positives. And then the off-label is sitting in the middle. It's not as perfect as the ground-truth positives, but it's not as bad as the ground-truth negatives. And I guess I just want to mention that having a high score is encouraging. Seeing all this separation was great, but we were really interested in looking into predictions and evaluating if they made biological sense. So this table is just showing us how the model actually looks. This table has some examples for 5 drugs. So we have a drug and we have the approved indication. And then, as I mentioned, we have access to a list of predictions. I will probably not have time to go through all these examples, but I wanted to show you one of them so we can understand what the model is doing. So I'll show you this example for aranidapine for Timothy syndrome. So aranidapine is a drug that is currently indicated for hypertension. It is an antagonist of the alpha-1 subunit of L-type calcium channel. And the Timothy syndrome is a rare genetic disorder that is caused by mutations in the same gene that encodes to the alpha subunit of the calcium channel. So yeah, this mutation basically results in heart-related defects, right? And what the model is telling us is that there is a mechanism overlap that is completely direct between aranidapine and Timothy syndrome. Essentially, we can see how the direct connection of aranidapine to the alpha subunits of the calcium channel is directly converging to the same mechanisms with Timothy syndrome. So really, the model is able to find this shared biology and identify that aranidapine is the top ranking prediction. I'm sorry, that Timothy syndrome is the top ranking prediction. But yeah, I think what I wanted to highlight is that we are not only having access to rankings that tell us that, hey, aranidapine is a good candidate for Timothy syndrome, but reusing the mechanistic paths from similar drugs, we have access to the explanations that, of course, make things more clear, and I think allow domain experts to understand the biology behind the predictions. All right, I think that's basically one of my last slides. I think I just want to wrap things up saying that drug repositioning is a faster and a cheaper way to find new treatments. Knowledge graphs allow us to connect biological information from different resources. There are a lot of graph representation methods out there, but one of the big limitations of them is that they are black box models. They give you a prediction, but they never explain you the biology behind. But with the strategy of our work, we frame the problem as a case-based reasoning problem, where we can use similar drugs and use them kind of like as a template to identify novel drug disease candidates. So that way we don't only have access to an score, but also to interpretable explanations. Some QRs if people are interested in looking into our code or read more details of this project. There are some technical parts and I guess more information about what happened in every step. So if people are interested in reading the manuscript, yeah, this is just a slide acknowledging that this project was done in collaboration with other teams. And of course, I want to thank my advisor, Professor Andrew Su, and a lot of other members that were also crucial in the development of this project.
Yes, thank you very much. There were some questions. Let's start with Alexander.
Hi, Carolina. Thank you for the talk. It's a great talk. I'm coming from dynamics modeling, so my question is probably because we're working with a PKPD model a lot, and how do we integrate this model into the three-step process that you have shown before? Thank you. Because we're working with the drug discovery, but so it can be also used in drug repositioning. So how do we, in what step we can use actually integrate this dynamics model into your three-step, yeah.
Yeah, that's a really interesting question, and I think it's part of the limitations of how knowledge graphs are modeled right now. So to my understanding, knowledge graphs are static, and we capture information in like a screenshot. So we know this information, and you capture it in the knowledge graph. So really capturing the dynamics or more complex information can be a challenge or a limitation. I believe there is like work trying to incorporate more dynamic or more more complex information into the knowledge graph. I think it's hard to me to answer where exactly we can incorporate it, because it's definitely a challenge that people are trying to tackle. Yeah.
Okay. Thank you so much. Thank you.
Jacob, you had a question.
Yeah, I'm trying to figure out the mechanics of the model. I'm a data scientist. I'm less concerned about all the biology stuff. I'm trying to figure out exactly what model are you training, and what's this embedding going on there? Like, what's the package? What are you training? How long does it take? These are things that interest me.
So basically, we train the graph neural network. So all the weights of the graph neural network need to be trained, because the model needs to understand what is the ground truth, what are the ground truth negatives, and I think this slide probably captures that in more clear. So at the end, what you are really training is, so you have your ground truth positives, and the idea is to make them more similar, right? So you start with random embeddings for all the nodes in your graph, and the graph neural networks allows you to make some transformations to end up with these embeddings, right? But you need to train them, and in a way that you train them is like you have ground truth positives. So you try to make ground truth positives more similar, and the ground truth negatives, you make them less similar to the ground truth positives, essentially. I think that basically is the summary of the training, relations, and so on. Yeah.
Let me ask it in my way. Perhaps it's like using terminologies is. So I'm familiar with graphs and graph databases. This is not what I'm asking. I'm asking you. I see several words that relate to other data science things. Like, for example, I see KNN. You can train a KNN network. Check it out from, take it out from, and put some fit. Like, you take it out and you put fit into it. Scikit-learn does that. You say embeddings. Embeddings have means layers in neural networks that you train. So you have all of those buzzwords here together, but they are detached from anything that I see. And when you see training process typically involves an algorithm like gradient descent. Not always. Sometimes it's something else. It's an optimization process. So I'm trying to figure out exactly what you're optimizing. If you're trying to optimize weights on your graph and you're running some algorithm on your graph, then I understand the graph is your model. Did I say it correctly?
So the graph is your input, right? If I understand the question correctly, and I think the objective, what you're trying to optimize is this similarity.
So I was going to try to understand this again. Also, I'm not so familiar with the graph neural network. So you start out with a feature space where you have a description of what the drug does, where you have a long vector that describes those behaviors. Now you're trying to do an embedding. What am I embedding in this situation here? I have a model of all of the graph associated with hypertension, all of the graph associated with bacterial infection. What are these points in my feature space? well, in my embedding space here that I'm representing.
So I guess you're asking like, what are these embeddings for hypertension or sorry,
Right.
or
What am I actually, what is being embedded there?
right. Okay. Yeah. So you start with a random embedding. I guess this is fine. Yeah. You start with a random embedding, and I guess what I'm trying to represent with this animation is that the embeddings or the GNNs capture the neighborhood. So essentially, every node has an embedding, and the transformations between one node and another captures that information. So, for example, acute coronary syndrome, the embedding is capturing information about the genes that are the neighbor for acute coronary syndrome. But at the same time, that this biological pathway also captures the information of their neighbor. So in this case, TNF, for example. So in some sense, the embeddings of every node captures information of how or what are the neighbors of that node, right? And GNNs, which are basically just transformation or like multiplications, allow us to get those embeddings. Ideally, the embeddings or for every node will capture the information of the neighborhood.
Your graph does computation with related to embeddings and weights. Did I say this correctly?
Right. Yeah, that's what the GNN does. Yes.
So now you run embedding and weights, and you run it through the graph, and you get an answer, correct? And then you apply a cosine similarity, and you are trying to get the numbers closer. So then you change either embeddings or weights or both.
Right. Yes. Essentially, yes.
So your model is the graph. Thank you. What algorithm are you using to optimize this, by the way?
We're using a relational graph neural network.
Oh, so you do have a neural network there. Now it
Yes.
makes sense.
Yeah, I guess I didn't explain it very clear. Apologize for that.
No, that's fine. You're speaking a different language than mine.
This audience tends to be more computational and mathematical, so they like to see all the details of what network architecture you use, how do you train it, what are the actual exact representations, and so on. So maybe we need to have you come back and give the technical paper that you had as well. But this is wonderful motivation.
Actually, I have some questions more about the benchmarking stuff. Maybe you have done something, but you didn't show that. And this drug repurposing or drug target interactions, they are very, very hot topic. And I wonder, you, of course, the advantage of your model is like you have this very rich information from a knowledge graph. However, how does your model compare to those kind of deep learning models that use this atomic information, like the molecular structures? That's another level of the information that used for drug target interactions. Were anything done with it?
Yeah, so I've seen like some knowledge graphs that incorporate the structure information of the drugs. We are not doing that, so we are trying to keep it more like on the mechanistic side of the information. So we're not capturing like what is the structure of clopidogrel. We're more like capturing how clopidogrel is affecting certain genes. But yeah, definitely you can incorporate the structure in the starting embeddings yeah, I think essentially that's how you could incorporate it. I guess it's a little bit different how people are modeling the structure in those models that you mentioned. But yeah, I guess my answer is just I've seen like people incorporating a structure of drugs or proteins in certain ways into the knowledge graph.
So another follow-up question is probably a bit technical, and because you have those hyperparameters, also for the KNN, you have these hyperparameters. So how does these hyperparameters affect your performance? Because for your case, I guess it's very hard to evaluate the performance because all the things are predictive, but you don't really have any validation to say whether the repurposed drugs is really gonna work, right? I mean, how did you do that, or how did you quantify the performances?
Yeah, no, that's a good question. But I guess I didn't explain it like correctly. But for validation, so we have the ground truth positives, the ground truth negatives, and the off-label. We do the classical separation of train, test, and dev data set. So some of the ground truth positives are never seen during training, and we evaluate them on test. And then the off-label data, it's also never seen during training, so we only use it for evaluation. And the off-label is really interesting because they are actually drug repositioning cases that are reported on the literature. So there are like drugs that have been repurposed for certain diseases. They are currently not approved by the FDA, but they are curated as novel drug repositioning cases. So we validate our model in that sense. So one of the metrics that we use is this hits at K that validates how well we are doing. So we have a drug and we have a ranking. We have like a ranking of possible diseases, and we evaluate how good we are doing for the ground truth positives. And as you can see here, we do better on the ground truth positives than the negatives one. So we optimize the hyperparameters in that sense. Yes, it can be hard, especially because the knowledge graph is really big. And I guess one of the challenges or one of the things that you have to balance on the knowledge graph is like how big you want your paths to be. Because I think I mentioned it over here, like when you collect information for a given drug and a given disease, the number of paths or the number of neighbors that you can consider can definitely be selected by you, right? So you can collect 100 paths or 10 paths, and that definitely can change the performance. So you have to find the balance between having a lot of paths or having just enough paths that are informative for your good performance. Because if you have a lot of paths, it can be really messy. But if you have very few paths, it's not informative. So you really need to find the balance of what gives you good information. That was mostly the challenging part. Like, how do we balance the information that we
that's
consider?
also very important, right? Yeah, because if you have too many possibilities, you have to prioritize and yeah, of course.
Yeah.
Another thing I would ask a bit more is like, once you have these predictions, you have the targets that can be used for repurposing. I don't know how likely this is. Of course, they are and these drug gene interactions. They are part. I mean, I don't know whether you use the really like a molecular information to validate, okay, whether this repurposed drugs or the predictive drug by you how can they dock together to make the effects, even though I know there are some database showing, okay, this drug can bind to particular genes, but in reality it's very, very challenging, right? So this really depends on the data or the database or knowledge you use in your knowledge graph, because this is really important matters. Even though the path exists, but probably the link or the association you see there is not really gonna work sometimes. I have to say like that. Yeah, that's the complexity of biomedicine, I guess. Yeah. Thank you a lot.
How did you account for class imbalance in your data? Because you had a different number of positive and negative training examples. Do you think that this affects the separation of patterns of classes when you do the embedding?
Yeah, that's a good question. I guess that's true. Like, how do we account with the ground truth positives and ground truth negatives? And I think it's hard, right? Because in biology or like for the drug discovery, we only have access to certain positives and certain negatives for a given drug. So we use it as it is. We don't really account for that imbalance. I guess the answer is that we are just using what we have as ground truth positives and ground truth negatives. Yeah. That's, I guess, the straightforward answer.
I was thinking that one of the things that, of course, you're dependent on having a very complete knowledge graph because you're not building the knowledge graph, you're using an existing knowledge graph.
Mm-hmm.
One of the things that the group here, and there was a question about how you could employ mechanistic models, dynamic mechanistic models in improving your training here. But I'm thinking about how to go the other way. Perhaps many people in this community build mechanistic dynamic models of complex systems. One of the hardest things we face is to select which components, what molecular species, what cell types, what biological mechanisms we need to include to solve a particular problem. And it seems that this is a method that might actually be very helpful for that, because we could say we want to build a model of disease X. We want to be able to relate it to drug Y. And then this will give us information from the knowledge graph about what mechanisms and components are most likely to be essential to building a mechanistic model. And so I think this could actually be quite helpful for us, because the hardest thing we face always is we can't model everything. We have to choose what components of the system we're going to model. That tends to be done in a rather ad hoc manner now. And this might allow the formalization of that kind of selection.
I think, yeah, we mostly focus on these explanation models. But yeah, no, but I guess it does make sense to model that in these networks.
Well, I don't think these networks by themselves are the dynamic models we're talking about. What I'm saying is when we build a dynamic model, we have to say we're going to include these pathways, these chemical species, these cell types, these interactions, these behaviors. Making the selection of what those things are that we are going to model dynamically at the moment is something that we do basically because of our experience. And if there were a more formal way to build what we would call conceptual models that we can then turn into computationally instantiable dynamic models, that would make our modeling much faster and much more consistent for us. And so I think that there could be a way here of identifying what the key features are that need to be included in our dynamical models.
Yeah, and I guess another way to see it is like you just incorporate everything and you allow a model to identify what are the most important components. I guess that could be another way to see it, because I can see that selecting the most important components to incorporate them all together can be challenged, as you mentioned, right? So maybe having a model that allows you to select the most important components could be a solution. But yeah, I see that could also be hard.
Right, because building these dynamic models is quite time consuming and validating them is also quite difficult. It requires a lot of experiments and it's expensive and time consuming. And so you want to build the simplest models that are dynamic and explanatory. So you don't want to include things you don't need. But we don't really have a good way of doing that at the moment. So I think this could be quite promising in that direction. I understand you're coming from a data science aspect of this, but I think that there are ways that this could apply in the world of mechanistic dynamic modeling that could be quite productive. John, that you had a question?
It's a really basic, one of my naive questions, like, what is the disease that we're talking about, any given? And so presumably there's some fundamental root cause of physiology that causes symptoms. The disease is perceived by the human or even the physician by its symptoms. So if I have a medication that treats disease A, and I go out and I say, what other diseases are defined by a similar set of symptoms? If I can find one, then maybe there's a reuse opportunity. But beyond that is the question of if the symptoms go away in disease B, like they did in disease A, were they different diseases at all? Or have neither of the medications even touched the root cause of the disease? Both of them affect the mechanism that causes the symptoms. And you haven't cured either one of them. Does that question make sense?
Right. Yeah. And that does make sense. And I think that can be captured in our model. So you're right, like diseases share similar symptoms, and therefore they can potentially be like if you have a disease that is treated by a certain drug, and then there is another disease with similar symptoms, maybe that drug can be reused. And yeah, that's right. We need to capture more complex information, not only the symptoms, because we also have like genes or the cellular components. And by this model, we can capture that information between diseases, and that way a given drug can be reused. In, I guess, the network that we have can be more complex than just symptoms or genes. Is more like the structure or the topology of the graph. And using the models, we can capture more complex information that can be really hard to see just by looking at names of genes or looking by the names of symptoms. So yeah, really the network which captures complex information can be helpful to analyze in a more like a comprehensive way, if that makes sense.
You know, fundamentally, are we treating symptoms or diseases? And every time we fail to do the mechanistic hypothesis for the statistical models, we might be making really bad mistakes.
I think your
Okay.
point, which is something maybe that is worth saying, is that you could imagine using a framework like this to ask the question, is a drug primarily ameliorating symptoms, or is it in fact solving the root cause of the illness? And that,
Yeah.
I think, is a kind of question what this method could be used for, and could be very interesting for that. Okay, I think we've had a good discussion today. Thank you so much. I'd love to have you come back and give us a more technical talk about some of the methodologies you've used. I think there'd be a good audience for that as well. Really appreciate you coming, and we will see everybody in 2 weeks. Thanks so much.
Thank you so much. I really enjoyed it.